And our final speaker for this section is Dr. Hiroaki Inoue, who received his PhD from Tokyo University of Agriculture and Technology after graduating from Teiko University. He is the technical manager for microbiology testing, Tsusuba Research Laboratory for Aquas Corp, and served on the working group for the editorial board for the guidelines for the prevention of Legionnaires disease. Thank you.
Thank you, Ms. Christine. Good afternoon. I'm Hiroaki Inoue from Aquas Corporation. I am very happy to have this opportunity. Today, I'm going to talk about consideration of the difference in results of PCR and plate culture method for detecting Legionella. What do you mean, difference in results of PCR and plate culture method? Doctors Wiley and Taylor have summarized the previous research, each detection rate like this. PCR is 72%, plate culture is 34%. These are based on the results of 28 research papers reported. PCR have more than 2 times higher the detection rate of the plate culture method. Why is the difference in detection rate of PCR and the plate culture? In the first place, what do PCR and plate culture methods detect? Of course, both methods detect Legionella. PCR detects a portion of Legionella gene. No matter whether Legionella are dead or alive. In contrast, the plate culture method detects only viable Legionella that colonize on the agar plates. Thus, Legionella can be detected by both methods, but these results have different meanings. PCR has problems. Primer specificity and inhibition of PCR. PCR is not a method that detects all Legionella genes with the same sensitivity. Primer selection is very important because the detection of Legionella and its sensitivity depend on the primer design. Also, PCR detects Legionella by amplifying target genes with DNA polymerase. In the presence of PCR inhibitors, may inhibit with PCR detection. So, it's necessary that confirm PCR inhibition by internal control. Plate culture method has also problems. The plate culture method has many testing steps, complicated and requires skill. Also, growth potential depends on the performance of the agar plates. Legionella detection is inhibited due to the growth of non-target microorganisms on the selective agar plates. As a result, the presence of Legionella may be underestimated. But the plate culture method is widely used because it's a standard method for Legionella testing. So, quality control is very important. This is the first page of ISO 11731, which is a detection method of Legionella by the plate culture method. It's written right here. Not all Legionella species are culturable. Therefore, the method described in this document do not recover all species of Legionella. In fact, the plate culture method is difficult to detect depending on the species of Legionella. I think the plate culture method was developed mainly to detect Legionella pneumophila. It's sudden, but let me show you about Legionella, because here is IDEX session. In fact, I have also evaluated Legionella. Handling of Legionella is very simple and easy. So, Legionella is a culture method that removes the difficulty of handling the conventional plate culture method. Legionella is a testing method to detect only Legionella pneumophila, but when evaluated on 132 public bathwater samples, it shows a higher detection rate of Legionella than the conventional plate culture method like this. The plots are shifted to the Legionella side. Based on these results, I think Legionella can be used effectively for testing bathwater. The state of Legionella in environments can be categorized into 3 types. 1, viable and can form colonies on agar plates. 2, viable but cannot form colonies on agar plates. 3, dead cells. They are illustrated in this figure. Plate culture can detect only viable Legionella that can form colonies. In contrast, PCR can detect Legionella all states. Then, the Legionella rate of PCR is higher than the detection rate of plate culture. In this case, the state 2 is called VBNC. The key point in interpreting the results of PCR and plate culture is how many these VBNCs are present. Next, how to detect VBNC Legionella. In nature, Legionella grow as a parasite on free-living amoeba. VBNC Legionella that do not colonize on agar plates can be detected using the amoebic co-culturing combined PCR method. We try to detect VBNC Legionella in the cooling tower water samples by the amoebic co-culture. Of 34 samples in which Legionella were detected by PCR and not detected by the plate culture. From 18 samples, 5 Legionella were detected by the amoebic co-culture. The amoebic co-culture can fill some of the difference in PCR and plate culture results. In conclusion, 1. The results of PCR and plate culture method cannot be simply compared because they have different meanings. 2. It's important to select the appropriate testing method for the purpose. Thank you very much.
Thank you, Dr. Inoue.